Okay, hopefully you all found my videos on my new YouTube channel because I couldn't use Jewel. So this content for today, Wednesday the 25th, will go up there. My goal is to post maybe 5 videos in one class period, 4 videos the next class period. I think that I may be a little bit behind, so I might post 2 or 3 today and then 1 tomorrow, but then we'll get caught up for the week. So ideally, we're looking at 9 to 10 of these little videos per week. We'll start today by hopefully finishing off neurotransmitters in Chapter 6. There are just a couple left to go through. And then we'll talk about Chapter 7, which is cell signaling. And so, I've said a few times already in the last few videos, well, this activates a G protein and then it does stuff. We're going to talk a lot more about how it activates that G protein, what that G protein then does in the cell in Chapter 7. So that's just a bit of where we're going. Okay, so let's get down to business today. We have to talk about now the category of neurotransmitters that we haven't talked about, these peptide neurotransmitters. If you remember, right, everything that we've mentioned before are small molecule neurotransmitters, typically a single amino acid or derivations of a single amino acid. These peptide neurotransmitters are typically chains of anywhere between 3 to 36 amino acids, so significantly larger in many cases than the small molecule neurotransmitters. Additionally, the way that they're synthesized is a lot different, right? So in neurons, pre-pro-peptides are synthesized, much like any other protein would be synthesized in a non-neuronal cell. So the synthesis begins as a pre-pro peptide. This is made in rough ER. And they're much, much longer than what our final version or what our final product is going to be. Okay. Then this pre-pro peptide is cut down a little bit into a propeptide and transported to the Golgi apparatus. So the propeptide version of this is then sent off to the Golgi. Whoops. If you remember your freshman year cell biology, remember that the Golgi can modulate or modify this particular propeptide. It might add some sugar molecules to it or something like that in order to glycosylate it. But there's typically some sort of stamp that's put on it that suggests where this propeptide is going to end up. On the transface of the Golgi apparatus, right, that's sort of the shipping face of the Golgi apparatus, the propeptide is packaged into vesicles. Right? And after packaging, there's further cleavage, modification of ends, etc. So this is where propeptides are then converted to the active peptides that we're going to know today. So in the vesicles, further cleavage of the propeptide takes place, modulation of their ends, phosphorylation, potentially even disulfide bond formation. A lot of final processing occurs in these vesicles once they're packaged up on their way out and down to axon terminals. And you can see what's happening in the diagram here to the right. So the pre-propeptide that's produced right in rough ER, just like we talked about. You can see how long it is. The signal sequence is eliminated and then moves on to the Golgi apparatus. So this propeptide is what makes its way to the Golgi. Here it's put into vesicles, right? And the vesicles are going to cleave this thing further to give us the active peptides that are going to be released, okay? Kind of interesting, propeptides are able to form more than one peptide and can form multiple different peptides. So propeptide, that says propeptide cleavage, can result in multiple different peptides. So if we take a look at our example here, right? Oh, come on. It's not liking my eraser. But what you can see here is you've got this active peptide, and it can be further cleaved potentially, right? So this could be the active peptide, or, right, a cleavage event could take place here, and what do we end up with are two separate and unique peptides. If we take a look at this example, right, here's our propeptide. And you can see after the cleaving events, right, what happens are these active peptides are sort of strewn or tied together. So each one of these, right, becomes the active enkephalin. What are the effects of propeptides? So let's look at effects. Effects really fall into any of 5 categories. So there are 5 categories of peptides: brain-gut, opioid, pituitary, and hypothalamic releasing. Well, that's only 4. Yeah, the 5th category, right, is basically everything else. So let's take a look at 2 examples, one that's brain-gut and one that's opioid. So before I talk about opioids, which I'll do next, I do want to mention substance P. So substance P is here. Whoa. Substance P is here, right? It's one of these brain-gut peptides, and you can see that it's 1, 2, 3, 4, 5, 6. What is it? 11 amino acids. So substance P. And I bring it up because it was the first discovered peptide neurotransmitter. So it's 11 amino acids. It's a potent hypotensive agent. So if you know this word hypotensive, right, it reduces blood pressure. Additionally, it has effects in the human gastrointestinal tract. So in addition to being present in high concentrations in areas like the hippocampus or the cortex of the brain, it's also used in the gut. So that gives it its identity as a brain-gut peptide. Basically, where do we find it? It's found in both places. Interestingly, the gene for substance P three other neuropeptides, or more, but 3 that we know of. Three other neuropeptides. So a great deal of diversity here, both in its signaling, right, that it can signal in the brain as well as the gut, and as well as that the gene can do a whole bunch of other, be a code for a whole bunch of other neuropeptides. Another neuropeptide that's gotten a lot of attention lately are opioids. And so these peptide neurotransmitters bind to the same receptor as the opium chemical. So these peptides, oops. They bind to the same receptor as opium, okay? And so we first discovered opioids in a search for endorphins, right? Basically endogenous compounds that look like morphine. So they were discovered when searching for compounds that look like morphine. So morphine is a plant alkaloid, basically this nitrogen-containing molecule that's the active ingredient in opium. Okay, so it's the morphine that exists within opium that binds to this particular receptor, and we know it has a real strong analgesic effect, right, that it can reduce pain. Many synthetic opioids have been synthesized, things like methadone and fentanyl. Right, so these are synthetic opioids in that they bind to the same receptor as morphine and some of these other endogenous chemicals that we'll talk about in a second. The problem with things like fentanyl is it binds and it's got about 80% of or 80 times, the potency. So it binds really, really tightly and it's highly, highly potent compared to any of the endogenous chemicals, right? The ones that our body produces. These things are exogenous and they're synthetic. Exogenous meaning that they're coming from the outside. And synthetic, they're not naturally occurring. We make them in a lab. Oops. Because we know what the structure of the receptor looks like and we know what morphine looks like. So morphine would be an example of an exogenous compound that's non-synthetic, right? It's naturally occurring. The search for endogenous compounds led to the discovery of what are called endorphins. Endo is an endogenous, orphan like orphine, morphine, right? So these are literally endogenous morphine. Right? These are your opioid neurotransmitters, right? And you can see here they make up a second family, and there are really three categories within this family, right? So there are the enkephalins, the endorphins, and the dynorphins, okay? 20 plus compounds so far that bind to these receptors that bind to opium, or the morphine portion of opium. So a lot of different types of compounds, a lot of diversity. They tend to be depressants. Whoops. Seems we're glitching out a little bit. And they modulate complex behaviors. I think we're going to stop here and pick up because I'm seeming to have some technical difficulties. So give me a minute and we'll be right back with the next video. I'm having trouble stopping, so I might just let it go for another minute. You can stop it now. I'm hoping that I don't have to remake the video, and that once we get to a minute, I'll be able to take control back here. It's not looking good.